Friends, today we will learn about bacterial genetics. Here we'll study about basic principles, synthesis of polypeptides, transcription, translation. Under synthesis of polypeptide, we'll study about transcription, translation, extrachromosomal genetic elements. Next is bacterial variation. Under bacterial variation, we will study about phenotypic variation and genotypic variation. Gene transfer. Under gene transfer, we will study about transformation, transduction, lysogenic conversion and conjugation. And next is genetic mechanisms of drug resistant, genetic engineering, DNA probes, polymerase chain reaction, that is in short PCR. Under this, we will study about principle, procedure and application of PCR. So, let's begin. Genetics is the study of heredity and variation. All hereditary characteristics are encoded in DNA. The chromosomal DNA characters constant from generation to generation. This is accomplished by most accurate replication of its DNA, yet heritable variations in small proportion of daughter cells occur. Now come to basic principles. DNA molecule is composed of two strands of complementary nucleotides wound together in the form of double helix as deduced by Watson and Crick. Bacterial nucleus contains a circular chromosome of a double-stranded DNA molecule of approximately 1000 micrometer, that is 1 mm long when straightened. Each strand has a backbone of deoxyribose, that is sugar, and phosphate groups. There are 4 nitrogenous bases, 2 purines and 2 pyrimidines. Purine means adenine and guanine, and 2 pyrimidine, that means thymine and cytosine. One of these 4 nitrogenous bases is attached to each deoxyribose. The two strands are held together by hydrogen bonds between the nitrogenous base on the opposite strands. This bond is such a specific manner that hydrogen bonds can only be formed between adenine and thymine and between guanine and cytosine. Adenine and thymine thus formed one complementary base pair and guanine and cytosine from another. Thus, when the arrangement of bases along one strand is TCGATC, the arrangement of the other strand will be AGCTAG. Double strand of this DNA will be like TCGATC, single bond AGCTAG. Hence, a molecule of DNA contains as many numbers of adenine as thymine and a guanine as cytosine. The ratio of A plus T to G plus C is constant for each species but varies widely from one bacterial species to another. During replication of DNA molecule, the two strands separate at one end, and each strand then acts as a template for the synthesis of a complementary strand with which it then forms a double strand. Now come to RNA. RNA is structurally similar to DNA except for two differences. That is, sugar ribose is present instead of deoxyribose, and the nitrogenous base uracil replaced thymine. There are 3 different types of DNA in a cell: messenger RNA, that is mRNA, in short mRNA, ribosomal RNA, that is rRNA, and transfer RNA, that is tRNA. Important definition in genetics. Let's know about the important definitions in genetics. Gene. First point is gene. What do you mean by gene? A segment of DNA that specifies for a particular polypeptide is called a gene. Again repeating, a segment of DNA that specifies for a particular polypeptide is called a gene. Now come to codon. What do you mean by codon? Genetic information is stored in the DNA as a code. Codon consists of a sequence of 3 nucleotide bases, that is, the code is triplet. Each codon specifies for production of a single amino acid, but more than one codon may exist for the same amino acid. Example, AGA codes for arginine, but CGU, CGG, CGA and AGG also code for the same amino acid arginine. This is also applicable to other amino acids as well. Now come to nonsense codons. What do you mean by nonsense codons? 3 codons like UAA, UGA and UAG do not code for any amino acid and act as stop codons for terminating the message for synthesis of polypeptide. These are called nonsense codons. Now come to synthesis of polypeptide. Let's know about synthesis of polypeptide. Genetic information in DNA is transcribed onto RNA and then translated as the particular polypeptide. Now come to transcription. What do you mean by transcription? A RNA polymerase attaches itself to the beginning of a gene on DNA and synthesizes mRNA. Using one of the strands in DNA as a template, this process is known as transcription. Since DNA acts as a template for synthesis of mRNA, therefore the bases in mRNA will be complementary to one strand of DNA. Now come to translation. The mRNA passes into cytoplasm and then mRNA and tRNA together on the surface of ribosome containing rRNA. The triplet base sequences on mRNA is known as codon. The base sequence on mRNA are recognized by anticodon sequences of RNA of tRNA. The tRNA molecule contains a triplet at one end and amino acid at the other end. The ribosome moves along the mRNA until the entire mRNA molecule has been translated into corresponding sequences of amino acids. This process is called translation. Now come to extrachromosomal genetic elements. Besides the chromosomal DNA, some bacteria may also be possess extrachromosomal DNA, which may be plasmids or episomes with chromosomal DNA of bacteria. Plasmids are circular DNA molecules and can replicate autonomously, that means independent replicants. It is often not possible to differentiate between the plasmids and episomes. Therefore, the two terms are frequently used synonymously. Plasmids and episomes are not essential for function of the host bacterium, but they may carry properties of drug resistance, toxicity, conjugation and others. Some plasmids have an ability to transfer themselves to other bacteria of the same and also of different species. These are called self-transmissible plasmids. This transfer occurs usually by method of conjugation. There are some other non-conjugative plasmids which cannot transfer themselves but can be transduced by their ability to transfer DNA from one cell to another. Plasmids have become important vectors in recombinant DNA technology or genetic engineering. Now come to bacterial variation. Let's know about bacterial variation. There are two types of variation. One is phenotypic variation, another is genotypic variation. The phenotype is the expression of various characters by bacterial cells in a given environment. The genotype is the sum total of the gene. It is the genetic constitution of all organisms. All the genes of the bacterial cell may or may not be expressed in a given environmental situation. Now come to phenotypic variation. Bacterial cell may exhibit different phenotypic appearance in different environmental conditions. Phenotypic variations are reversible. Some examples of environmental influence on bacteria are synthesis of flagella, synthesis of enzymes. Let's know about synthesis of flagella. Salmonella typhi, that is typhoid bacillus, is generally flagellated, but when grown in phenol agar, the flagella are not formed. This effect is reversed when subcultured from phenol agar into broth. Now come to synthesis of enzyme. E. coli possesses the genetic information for the synthesis of the enzyme beta-galactosidase, but the actual synthesis takes place only when grown in a medium containing lactose. This enzyme is necessary for lactose fermentation. Such enzymes, which are synthesized only when induced by the substrate, are called inducible enzymes. The enzymes which are synthesized irrespectively of the presence or absence of the substrate are called constitutive enzymes. Now come to gene regulation, that is operand concept, by Jacob and Monod. Lactose fermentation of E. coli requires three enzymes: beta-galactosidase, galactoside permease, and transacetylase. These enzymes are coded by the structural genes LAC-Z, LAC-Y, and LAC-A respectively. These genes are arranged in sequence forming a functional unit, LAC operand. The regulator gene is LAC-1, which codes for the repressor, which is an allosteric protein molecule. This repressor can combine either with the operator region or with the inducer. The promoter and the operator regions lie between LAC1 and the structural LAC genes. The RNA polymerase has to attach to promoter region and travel along the structural genes in sequence for the transcription of RNA for enzyme synthesis. In the resting stage, the repressor molecule is bound to the operator gene region, preventing the travel of RNA polymerase from the promoter region to the operand. The repressor molecule has also affinity. The repressor molecule also affinity for lactose, in the presence of which it leaves the operator free. So that transcription can take place. When the lactose is completely metabolized, the exhausted, the repressor again attaches to the operator, switching off transcription. Lactose acts both as the inducer and the substrate for enzyme. Now come to genotypic variation. These variations occur in the genetic material. These are stable, heritable, and non-influenced by the environment. Genotypic variations may occur in bacteria by mutation or gene transfer. Now come to mutation. Mutation is a random, undirected, heritable variation caused by change in the nucleotide sequence of the DNA of cell. It can involve any of the numerous genes present in bacterial chromosome. The frequency of mutation ranges from 10 to the power minus 2 to 10 to the power minus 10 per bacterium per division. Mutation occurs spontaneously, but its frequency may be enhanced by mutagens such as UV rays, alkylating agents, 5-bromouracil, and acridine dye. Mutation is a natural event taking place all the time. In all dividing cells, most mutants go unrecognized as the mutation may be lethal or may involve some minor function that may not be expressed. Mutation is best appreciated when it involves a function which can be readily observed by environmental methods. For example, E. coli, Escherichia coli mutant that loses its ability to ferment lactose can be readily detected by an McConkey agar. Mutation can affect any gene and hence may modify any characteristic of the bacterium. Example, sensitivity to bacteriophage, loss of ability to produce capsule or flagella, loss of virulence, alteration in colony morphology, pigment production, drug susceptibility, biochemical reactions, antigenic structure. The practical importance of bacterial mutation is mainly in the field of drug resistance and the development of live vaccines. Some mutations involve vital functions, producing non-viable mutant, that is lethal mutation. An important type of lethal mutation is conditional mutations. A conditional lethal mutant may be able to live under certain conditions but not under restrictive or non-permissible conditions. One example of such type of mutant is the temperature sensitive mutant, which is able to live at the permissible temperature, say 35 degrees centigrade, but not at the restrictive temperature, that is 37 degrees centigrade. Types of mutation. Now let's know about types of mutation. There are broadly two types of mutation. One is point mutation, another is multi-site mutation. Let's know about point mutation. It may occur by base pair substitution and frame shift mutation. Now let's know about base pair substitution. A single base pair has been substituted for the another pair. It can be subdivided into transition and transversion. Now let's know about transition. This happens by replacement of one pyrimidine by another pyrimidine and one purine by other purine at replaced by GC. Transition is the most frequently occurring mutation. Transition is the most frequently occurring mutation. Now come to transversion. When a purine is replaced by pyrimidine and vice versa, it named transversion. Example, GC changes to CG. Now come to frame shift mutation. Sometimes during DNA replication, one or a few base pairs are inserted into or deleted from the DNA. This shifts the normal reading frame of the coded message, forming new set of triplet codon. The coded message is read correctly up to the point of addition or deletion, but the subsequent codons will specify the incorrect amino acids. Now come to multi-site mutations. Large number of base pairs are altered in DNA. This may be of four types, that is addition, deletion, duplication and inversion. Other types of mutations. Let's know about the other types of mutation. What is induced mutation? What do you mean by induced mutation? Although mutation occurs spontaneously, its frequency is greatly enhanced by certain agent called mutation. These may be physical or chemical agents. Now come to physical agents, that is UV light, ionizing radiation, example X-rays, visible light, heat. Chemical agents, first is 5-bromouracil, second is 2-aminopurine, third is nitrous acid, fourth is acridine dye. Now come to nonsense Mutations. Nonsense mutation. When a nonsense codon, that means UAG, UAA or UGA, is found within a gene by mutation, the protein synthesis is terminated prematurely and a partial peptide is produced during translation. This is called a nonsense mutation. Now come to missense mutation. Both substitution and frame shift mutation give rise to an altered codon which specifies a different amino acid from that normally located at a particular position in the protein. This is called missense mutation. Now come to suppression mutation. A mutation that restores the function of a gene altered by previous mutation is called suppression mutation. Now come to demonstration of mutation in laboratory. Mutation can be recognized by both gene sequencing as well as by observing phenotypic changes. Fluctuation test and replica plating methods are useful for demonstration of large number of mutant colonies. Now come to fluctuation test. Luria and Delporg in 1943 demonstrated that bacteria undergo spontaneous mutation independent of environment. They studied bacteriophage sensitive Escherichia coli samples from small volume cultures and a single large volume culture were plated on solid media containing bacteriophage. The mutant bacteria formed colonies. Colony count was compared. There was wide fluctuations in the number of bacteriophage resistant colonies in small volume cultures as compared to a single large volume culture. On statistical analysis, it was observed that mutation occurred randomly, some early and some late, which lead to wide fluctuation. However, in large volume culture, fluctuations were within limits. This experiment was not widely appreciated, probably due to the complicated statistical evaluation. It was a simple but elegant replica plating technique that proved the point beyond doubt. Now come to replica plating method. Lederberg in 1952. Colonies of bacteria were transferred for master plate onto a number of other plates using a velvet template. Relative position of all the colonies was retained by replica plating on culture plates with and without bacteriophages. They were able to demonstrate that bacteriophage resistant mutants appeared without ever having had contact with the selective agent. Now come to gene transfer. What do you mean by gene transfer? Various methods of gene transfer are as first is transformation, that means uptake of naked DNA. Second is transduction, that is through bacteriophage. Lysogenic conversion and conjugation, that is plasmid mediated. Now come to first point, that is transformation. Transformation is the transfer of genetic information through the free or naked DNA. In 1928, Griffith found that mice died when injected with a mixture of live non-capsulated, that is raw, no capsule, pneumococci derived from capsular type 2 and heat killed, that is smooth capsulated strain from type 1, neither of which separately proved fatal. The live type 1 capsuled pneumococcus could be isolated from mice that died. It showed that raw forms, that is non-capsulated, were transformed into the smooth forms. It demonstrated that some factors in heat-killed type 1 pneumococcus had transferred the information for capsule synthesis in the non-capsulated strain. Later on, this transforming factor was identified as free DNA by Avery, MacLeod and McCarthy in 1944. Transformation has been studied mainly in bacteria like pneumococci, H. influenza and Bacillus species. Now come to transduction. Transmission of a portion of DNA from one bacterium to another by a bacteriophage is known as transduction. Bacteriophages are viruses that parasites bacteria or multiply in it. During the assembly of bacteriophage progeny inside infected bacteria, errors may happen occasionally. Besides its own nucleic acid, host DNA may accidentally be incorporated into the bacteriophage. This is known as packaging error. Hence, when this bacteriophage infects another bacterium, host DNA is transferred and the recipient cell acquires new characters encoded by donor DNA. Now come to types of transduction. First is generalized, second is restricted. First, let's know about generalized transduction. Transduction may be generalized when it involves any segment of donor DNA at random. Second is restriction transduction. Transduction may be restricted when a specific bacteriophage transduces only a particular genetic trait. It has been studied intensively in the lambda phase of E. coli. Now come to role of transduction. Transduction is not only confined. Let's know about role of transduction. Transduction is not only confined to transfer of chromosomal DNA, but episomes and plasmids may also be transduced. Penicillin resistance in staphylococci is due to plasmids transferred from one bacterium to another by transduction. Transduction provides an excellent tool for the genetic mapping of bacteria. Any bacteria for which bacteriophage exists can be subjected to transduction. Transduction has also been proposed as a method of genetic engineering in the treatment of some inborn metabolic defects. Now come to lysogenic conversion. Bacteriophages have two types of life cycle inside the host bacterium. First is virulent or lytic cycle. Second is temperate or non-lytic cycle. Now come to virulent or lytic cycle. Large number of progeny phages are formed and subsequently these progeny phages are released, causing death and lysis of the host cell. Now come to temperate or non-lytic cycle. In contrast to virulent cycle, the host bacterium is unharmed. The phage DNA remains integrated within the bacterial chromosome as the prophage, which multiplies synchronously with bacterial DNA. The prophage acts as an additional chromosomal element which encodes for new characters and it transfers to the daughter cells. This process is known as lysogeny or lysogenic conversion, and bacteria harboring prophages are known as lysogenic bacteria. In lysogenic conversion, the phage DNA transfer is the new genetic element in contrast to the transduction where the phage acts only as a vehicle carrying bacterial genes. Now come to example of lysogenic conversion. Diphtheria bacillus produces toxin and therefore virulence only when lysogenization with the phage beta occurs. Elimination of phage from toxigenic strain renders it non-toxigenic. Now come to conjugation. The transfer of genetic material from one bacterium to another by mating or contact is called conjugation. Donors are those bacteria that contain F plasmid, while cells lacking F plasmid act as recipients. F plasmid is conjugated plasmid which encodes for sex pillars necessary for conjugation. This plasmid is also named as sex factor or fertility factor. When other similar plasmid were also discovered, a name of transfer factor was given to all such plasmids which conferred on their host cells the ability to act as donor or male cells in conjugation. Bacterial conjugation was first discovered by Lederberg and Titam in 1946 in Escherichia coli K12 strain. E. coli K12 strain. Now come to during conjugation, the plasmid DNA replicates and copy of it passes from donor to the recipient cell, probably along the sex pillars, that is conjugation tube. As a result, recipient becomes donor F minus and F plus. F minus is the recipient, F plus is the donor, and can in turn conjugate with other female cell, that is F minus. This character of maleness, F plus, in bacteria is transmissible or infectious. Now come to HFR cell and F cell. F factor. F factor of plasmid has the ability to exist in some cells in the integrated state with the host chromosome, that is same as episome. These cells are able to transfer chromosomal DNA to recipient cells with high frequency, therefore named as HFR cells. Conjugation with an HFR cell, an F minus rarely becomes F plus, but it receives chromosomal DNA from the donor. The conversion of an F plus cell into an HFR cell is reversible. When the F factor reverts from the integrated to free state, it may be sometimes carry with it some chromosomal DNA from adjacent site of its attachment. Such an F factor carrying some chromosomal DNA is named as F factor. When F dash cell conjugate with a recipient, it transfers along with the F factor the host DNA incorporated with it. This process is called sex duction. Now come to Cholecogenic factor and resistant transfer factor are two medically important factors which can be transferred by conjugation. Now come to cholecogenic factor. Some strains of coliform bacteria produce colicins, which are antibiotic-like substances lethal to other enterobacteria. Bacteria other than coliforms also produce similar kind of substances. Example, pyocin by Pseudomonas pyoceanae, diphtheria by Corynebacterium, diphtheria. Hence, a general term bacteriocin has been given to these substances. These bacteriocin are useful for intraspecies classification of certain bacteria. Colicin production is encoded by a plasmid called the cofactor, which may be transferred by conjugation. Now come to resistant transfer factor, RTF. This plasmid is responsible for the spread of multiple drug resistance among bacteria. It was first reported by Japanese workers in Shigella strains resistant simultaneously to 4 drugs. They observed that patients excreting such Shigella strains also shared Escherichia coli strains resistant to some same drugs. Transfer of multiple drug resistant from E. coli to Shigella strain was demonstrated. The resistance is plasmid mediated and is transferred by conjugation. This mechanism of drug resistance is known as transferable or infectious drug resistance. Resistant transfer factor and the resistant determinant are two components of plasmid known as an R factor. The RTF is responsible for conjugational transfer, while each R determinant carries resistance for one of the several drugs. An R factor can have several R determinants, and resistance to many drugs can be transferred simultaneously. Sometimes RTF dissociates from R determinants and both exist as separate plasmids. In such cases, the resistance is not transferable, though the host cell remains drug resistant. The RTF can also attach to its determinants other than those for drug resistance. Enterotoxin and hemolysin production in some enteropathogenic Escherichia coli are shown to be transmitted by this transfer factor. Now come to transferable drug resistance is seen in various pathogenic and commercial bacteria of man and animals such as E. coli, Klebsiella, Proteus, Vibrio, Pseudomonas. Transfer of drug resistance occurs readily in vitro, but in the normal intestines it is inhibited by several factors such as anaerobic conditions, bile salts, alkaline pH, and abundance of anaerobic bacteria. But in the intestine of persons on oral antibiotic therapy, transfer occurs readily due to the selection pressure produced by the drug. Now come to transferable drug resistance. Transferable drug resistance involves all antibiotics in common use. Bacterial carrying R factors can be transmitted from animals to man. Hence, indiscriminate use of antibiotics in animals or in animal feeds can lead to an increase of multiple drug resistance in the community. Now come to genetic mechanism of drug resistance. Bacteria may acquire drug resistance by mutation or by one of the methods of genetic gene transfer. In clinical practice, mutational resistance is very important. In tuberculosis, if a patient is treated with streptomycin alone, initially organisms die in large numbers, but soon resistant mutants appear and multiply unchecked. Transferable drug resistance mediated by the R factor is an important method of drug resistance. Acquisition of an R factor simultaneously confers resistance to several drugs. Therefore, there is no use of combined therapy. Now come to resistant gene transfer by transformation can also be demonstrated experimentally, by its significance in nature is unknown. The plasmid carrying the gene for beta-lactamase production can be transferred from a penicillin-resistant Staphylococcus to a susceptible Staphylococcus by transduction. Now come to transposable genetic elements. Transposable genetic elements are specific sequences of DNA segments that have the ability to move from one plasmid to another plasmid or from plasmid to chromosome and vice versa, and also within the chromosome. Because of their ability to insert into many sites both on plasmid and chromosome, they have assumed the popular name of jumping genes. The transfer of genetic material from one DNA molecule to another is called transposition. It does not require any DNA homology between transposable element and the site of insertion. It is therefore different from recombination. Transpositions are larger, that is 4 to 25 kilobase per kb. Transposable genetic elements and contain additional genes that encode for at least one function. They may also encode for toxin production. Transposon is defined as a segment of DNA with one or more genes in the center and the two ends carrying inverted repeat sequences of nucleotides. Nucleotide sequences complementary to each other but the reserve order because of this feature. Each strand of the transposon can form a single stranded loop carrying the gene or genes and a double-stranded stem formed by hydrogen bonding between the terminal inverted repeat sequences. Small transposon, that is 1 to 2 kilobase pair, are known as insertion sequences or IS elements. Transposons attach at certain regions of chromosomal plasmid or phage DNA. Bacteria acquire new characters by the insertion of transposable elements. Unlike plasmids, transposons are not self-replicating and are dependent on chromosomal or plasmid DNA for replication. It has been suggested that our plasmids may have evolved as collections of transposons, each carrying a gene that confers resistance to one or several antibiotics. Transposition is a mechanism of amplifying genetic transfers in nature. Now come to genetic engineering. By genetic engineering or recombinant DNA technology, it is now possible to isolate the gene coding for any desired protein from microorganisms or from cells of higher form of life including man, and to insert them into suitable microorganisms in such a way so that it can be expressed in the formation of specific protein. Such cloning of genes in microorganisms enables the preparation of desired protein in pure form and in large quantities at a reasonable cost. The DNA can be cleaved by enzymes called restriction endonucleases, and the fragments containing the desired gene are isolated. These desired DNA fragments are covalently bound to vectors or carriers such as plasmids or temperate bacteriophages. The recombinant DNA molecule is sealed by an enzyme, DNA ligase, and is then introduced into a bacterial host. Usually E. coli. By transformation, many other bacteria and yeast have also been employed for introduction of recombinant DNA molecule. Escherichia coli containing recombinant molecule is grown on a suitable medium and bacterial colonies carrying specific genes are produced. Thus, desired protein is obtained in large quantities by growing this E. coli. Now come to application of genetic engineering. First point is production of vaccines. Preparation of certain vaccines is done by producing specific antigen against which antibody is required. Example, hepatitis B, rabies and B pertussis vaccines. Now come to production of proteins. Genetic engineering has also been used for the production of proteins of therapeutic interest. These include human growth hormone, insulin, interferons, interleukins-2, tumor necrosis factor and factor 8. Now come to gene therapy. Genetic diseases can be cured by introducing normal genes into the patient. Now come to restriction endonucleases. These are microbial enzymes which split DNA into fragments of varying lengths. They cleave double-stranded DNA at specific oligonucleotide sequences. Many such enzymes, for example EcoR1, HIND3, which act at different nucleotide sequences, have been recognized. The natural function of these enzymes in bacteria may be destruction of foreign DNA which may enter the bacterial cell. Now come to DNA probes. DNA probes are radiolabeled or chromogenically labeled pieces of single stranded DNA which can be used for the detection of homologous DNA by hybridization. Hybridization is the technique in which two single strands of nucleic acid come together to form a suitable double-stranded molecule. All microorganisms contain some unique sequences of nucleic acid within their genome that distinguish them from all other organisms. This unique sequence of nucleic acid can be recognized by hybridization with a DNA probe. Various diagnostic DNA probes have been developed for identification of different microorganisms. Probes containing sequences unique to the microbe can be added to microbial cultures, body fluids, or other specimens suspected to contain the microbes or its DNA. The DNA probe hybridizes with the complementary sequences on the microbe's DNA. The advantages of DNA probes are either high degree of specificity and ability to detect minute quantities of DNA. They are also very useful in diagnosis of microbes that are either difficult or impossible to culture. DNA probes which bind to RNA can also be designed and has been particularly used to locate ribosomal RNA. Now come to application of DNA probes. Applications of DNA probe technology we will now discuss. Diagnostic DNA probes are recognized Legionella, Pneumophila, Campylobacter jejuni, M. tuberculosis, Helicobacter pylori, Escherichia coli, that is LT and ST toxins, hepatitis B virus, Plasmodium falciparum, rotavirus, and human immunodeficiency virus have been developed. Now come to blotting techniques. DNA fragments are obtained by restriction enzyme digestion and separated by gel electrophoresis. These fragments can be transferred from the gel by blotting to nitrocellulose or nylon membranes that bind the DNA. The DNA bound to the membrane is first denatured and then hybridized with radioactive single-stranded DNA probes. Hybridization results in formation of radioactive double-stranded segments which can be detected on X-ray film. This technique of identifying DNA fragments by DNA hybridization is called Southern blotting after E.M. Southern, who developed it. This is a highly sensitive technique. A similar procedure for analysis of RNA has been called Northern blotting. Here the RNA is separated by gel electrophoresis blotted and identified using labeled probes. A technique for identification of proteins is called immunoblotting. The Western blot test is one example which has been used as confirmatory test for the diagnosis of HIV antibody in sera. Now come to polymerase chain reaction, PCR. In short, PCR, polymerase chain reaction, was first developed in 1983 at Cetus Corporation, USA. It has made a revolutionary impact in molecular biology. In 1993, Carey Mullis was awarded the Nobel Prize for this work. Now come to let's know about the principle. It is a DNA amplification system that produces a large amount of DNA in vitro from small amounts of starting material. It amplifies a specific DNA sequence of interest. That means it may be DNA or maybe gene. Now come to know about procedure. First procedure, denaturation, primer annealing, and DNA synthesis and detection of amplified product. First, denaturation. The double stranded DNA is dissociated to a single stranded DNA at 94 degrees centigrade, that is denaturing temperature. Second point is primer annealing. The temperature is produced to 50 to 60 degrees centigrade, then oligonucleotide primers attached to target DNA. This temperature is called annealing temperature, and the process is known as annealing of primers. Third point is DNA synthesis. Polymerase enzyme derived from Thermus aquaticus in short TAC triggers the formation of new DNA strand from pre-nucleotides. TAC polymerase and nucleotides are added in the tube for formation of new strands of DNA. These three steps are repeated again and again, a process that is automated by the thermocycler or PCR machine. The products of first cycle become the template for the next cycle. After 20 to 30 cycles or more, the exponential increase in the amount of DNA occurs. Now let's know about fourth point, that is detection of amplified product. Amplified DNA can be detected by gel electrophoresis. And now let's know about the application of PCR. The PCR provides extremely rapid analysis in one day. PCR is a versatile tool useful in infectious, genetic or neoplastic diseases, in forensic investigations, and in the examination of phylogenetic relationships in evolution. It has been applied in clinical laboratory for diagnosis of various infectious agents. A specific DNA sequence of a particular infectious agent is amplified with the specific primers. Finally, I have finished this portion. Any questions regarding this, you can ask me in the comment box, and subscribe my channel to know more updates about biotechnological topics. Thank you.